Okay guys, in this lesson we are going to focus on the control mechanisms involved in cellular metabolism. So the first thing that we're really going to talk about are enzymes, because we should all know that enzymes play a huge role in controlling chemical reactions. In addition to them being responsible for breaking down substances, they're also just a huge control molecule. They are a specific type of globular protein, and the word globular is really just based on a globe shape. Alright, so it's really just based on the shape of the protein, and that name is one of the two main classes of proteins that exist. I don't want you guys to focus too heavily on this, but just so that you know, globular proteins are named based on their shape. Like I said, they control rates of chemical reactions, and we should know that many enzymes have the suffix ase. So like lactase, which we've talked about with lactose intolerance, lipase, protease, and you can see that they're kind of named after the substrate, the substance that they're actually designed to act on. So protease being for protein, lipase being for a lipid, lactase being for lactose, and we just have that ase as our suffix. The way that enzymes work is that they actually lower the energy that's needed to start the reaction, and that energy is called the activation energy. So what actually happens is that the enzyme plays a role in making less energy necessary in order for the reaction to be started, so the reaction is going to kickstart more quickly and occur more easily. Another thing to point out is that enzymes don't get consumed by the reaction. Because they're not consumed, they're only needed in very small quantities. So we don't need large amounts of enzymes. We only need small amounts because they're not being consumed and disposed of. They also have a specific shape, and that specific shape is referred to as being substrate specific. And I drew you guys a picture basically showing you that if this was the enzyme, then the substance that it was meant to act on would be a perfect fit for that lovely little shape. Again, sorry about my drawings, guys. But that's called substrate specific. So obviously that substance is going to fit in place, whereas a molecule that's shaped like this is not. So this is just kind of a picture showing you exactly what I'm talking about, that you have a particular shape of the enzyme, and the molecule is going to be meant to fit specifically in there. So in this case, what we actually have is the enzyme, which you see in purple, and we have two separate substrate molecules. And what a substrate is that the substrate is the molecule that the enzyme is meant to act upon. So the molecule that the enzyme will act on. So in this case, we have two separate substrate molecules. And what they're going to do is they're both going to come in, and together they're going to perfectly fit into what's called the active site. Right? The active site is the part of the enzyme where the substrate fits. So it's the location on the enzyme where the substrate fits. And as you can see, these two substrate molecules are going to fit in place, and they're actually going to become one. While they're bonded together, it's called an enzyme substrate complex. And then what actually happens is that the product molecule is going to be released, and the enzyme remains unaltered, completely unchanged, and that's because, like we said before, it's not actually consumed by the reaction. So in this case, we have two small molecules, and they combine to form a large molecule. And we should know that in that case, that's going to be our synthesis reaction. And in our synthesis reaction, we should know at that point that what we're really talking about is anabolic reactions, right? We're talking about building up. It's an anabolic reaction in this particular picture. Now, another thing to point out is the fact that metabolism isn't just like one single reaction occurring at a time. It's really a series of reactions. So immediately the product of one reaction is going to lead directly into the next reaction. And it's kind of going to be a chain of reactions occurring to get the final product that's needed. So rarely is it just one simple reaction. So as in our picture, we show that we have one substrate that the enzyme will act on to get our second substrate. That one is going to be altered by another enzyme to get the next substrate, and so on and so forth. So it's really kind of like a chain reaction that occurs. And like I said, each new substrate is a product of the previous reaction, and it's going to be the reactant for the future reaction. So they just kind of continue on in series. But we have these rate-limiting enzymes in place to kind of help regulate the metabolic pathways, these series of reactions. What that basically means is that certain enzymes, there's going to be a limited number of them. It's usually early on in a series, and that's really to ensure that you don't have a buildup in the middle somewhere, because what will happen is that somewhere a product will start to accumulate if it can't move on in the reaction. So it's typically just one of the first enzymes that there's a limited number of, and that limited number kind of slows down the process and keeps it in check. So basically it doesn't matter if the substrate concentration increases, because after a certain point the enzyme is basically maxed out. So if enzyme A, if there's only, let's say, 3 of enzyme A, then it really doesn't matter if there's 16 of substrate 1, because only 3 are making it through. You know, you're only going to be able to use as many as you have enzymes to work with. So it kind of controls that metabolic pathway and keeps it in check and makes sure that it's occurring at the proper rate and not too fast, as well as not too slow, you know, making sure that there's a proper number of enzymes needed for life processes. There are also other factors in place to kind of help keep metabolic processes in control, and those can be cofactors and coenzymes. So what they're actually used for is to actually trigger enzymes and make the enzymes do what they're supposed to do. A cofactor can be an ion or a coenzyme, so really a coenzyme is a specific cofactor, right? So they're all really cofactors, but you can be more specific and talk about coenzymes. And what actually happens is that sometimes cofactors can help active sites achieve their proper shape. They can make sure that the active site actually fits properly with the substrate, or they can actually be used to help bind the enzyme and substrate together so that the reaction, the enzyme-substrate complex, can exist and a reaction can take place. Coenzymes, this specific type of cofactor, are oftentimes vitamins. And vitamins are actually something that come from diet. Vitamins are from diet. And that's really because there are these organic molecules that we can't synthesize enough of, sometimes not at all, in order for our body to properly function. So these cofactors kind of help being a missing puzzle piece to make sure that the reaction takes place. And the last thing that we're going to talk about for controlling metabolic pathways is what happens when the metabolic pathway isn't properly controlled or is altered in some way by an outside force. And what actually happens is denaturation, this process where a protein loses their conformation. And you guys should remember that word conformation. What we're really talking about is that specific shape. When we talked about proteins back in our biochemistry chapter, we talked about primary structure, secondary structure, tertiary structure, and quaternary structure of proteins, and talking about those different structures are called the conformation of the protein, and they're responsible for giving it its specific shape. And that specific shape is directly related to the function of the protein. So if they lose their conformation, as a result, they lose their functionality, which is a huge problem, because if they lose their functionality, then they can't do what they're supposed to do. Some outside elements that can actually denature proteins and enzymes are heat, radiation, electricity, chemicals, pH. So a really good example, I gave this to you guys in class, was talking about fevers. Fevers being a condition where denaturation of enzymes is something that needs to be recognized. Some enzymes they actually start to denature at around 106 degrees Fahrenheit, which is why we have these thresholds for seeking immediate medical attention. If those enzymes stop functioning, depending on what enzyme it is, it can be life-threatening. Many are actually denatured at about 113 degrees Fahrenheit. So when you start getting up and closer to 113, more and more of these enzymes and these proteins are going to become denatured and stop doing their jobs, and it's going to become a problem for the body. And then all of them will be denatured at 131 degrees Fahrenheit, which I'm sure we would all know that if all of your enzymes and all your proteins are becoming denatured, you're probably dead at that point. Easy way to think about denaturation is thinking about egg whites, which naturally are not actually white. They're transparent. They're that runny, clear substance when you crack an egg. That's the albumin, the protein that's in the egg white. And what actually happens is when you heat it, when you provide that outside factor and you heat it, you denature that protein, the albumin. And when you denature it, it turns white. So that's why when you cook eggs, the clear substance starts to turn white. It's because you're actually denaturing the proteins that are in that egg. Alright guys, that pretty much wraps it up for our information on controlling metabolic processes. If you have any questions, please feel free to see me.